This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy. We go deep and lightly surface the big ideas.
So I want you to just imagine for a moment that you are walking down a totally quiet street. It's springtime.
Right, nice weather, maybe a gentle breeze.
Exactly. The gentle breeze picks up, and it carries with it this sweet, very unmistakable scent of cherry blossoms.
Which, for most people, is a great thing.
Yeah. I mean, it triggers feelings of calm, maybe memories of a spring festival, or just, the simple pleasure of a nice day out. But for you, the second that scent hits your olfactory receptors, your body just goes into absolute overdrive.
Like a full-blown panic response.
Totally. Your heart rate skyrockets to like 140 beats per minute. Your palms are sweating. Your pupils dilate. This profound, just completely inexplicable sense of terror washes over you, and your fight or flight response is fully activated.
You're basically having a panic attack right there on the sidewalk.
Right. You're having a massive panic attack. But here's the really strange part about all of this. You have never, ever had a bad experience with cherry blossoms in your entire life.
You might not have even seen a cherry blossom tree before in person.
Exactly. But your grandfather did. And your grandfather? Well, he learned to be terrified of that smell.
It honestly feels like an error in the system, right? Like you are experiencing a memory that just straight up does not belong to you.
Yeah, it's an adaptation to a threat that you have never personally encountered. You're panicking over a flower because your biology has this ghost in the machine.
Which is fascinating because for the longest time, science essentially told us this scenario was completely impossible.
Right, because we are all taught in basic biology that inheritance is this strict one-way street of static code.
The blueprint. You get half your DNA from your mother, half from your father, and that is it.
Right. And if you break a bone, or you learn a new language, or you survive some horrific trauma, those experiences are yours alone. They end with you.
That was the absolute dogma.
But the massive stack of sources we are pulling from today for this deep dive, I mean, we are looking at landmark studies in Nature Neuroscience, decades-long cohort studies on historical famines, and even some really wild recent data on the legacy of the COVID-19 pandemic.
And all of it suggests something entirely different than what we were taught.
Completely different. We are looking at the absolute frontier of what's called transgenerational epigenetic inheritance. So our mission today is to explore how the actual lived experiences of our ancestors are physically written into our biology.
We want to look at how those marks are made, how they are surprisingly transient, but sometimes become part of our ongoing evolutionary fabric, and crucially, how they can actually be erased.
Exactly. And what all of this means for the evolutionary relay race that, we are all participating in right now.
To really grasp the magnitude of this shift, though, I think we have to look back at the rigid firewall that defined biology for well over a century.
Weismarier.
Right, the Weismann barrier. It was named after Argus Weismann, who is this 19th century evolutionary biologist. And he proposed this strict, impenetrable separation between somatic cells.
Which are the cells making up your brain, your heart, your skin, right?
Exactly. He proposed a separation between those somatic cells and the germ cells, which are your sperm or your eggs.
So the idea being that your somatic cells, they can go out and experience the world.
Yeah.
They can get damaged by the sun, they can build up muscle from lifting weights, or they can be altered by intense trauma.
But that information is permanently trapped. It can never, ever cross over into the germline.
The Weismann barrier was basically the ultimate biological quarantine. It dictated that the slate is wiped perfectly clean with every single generation.
You inherit the raw genetic sequence, the DNA, but none of the actual life experiences of the organism that carried it.
And that was the bedrock of classical genetics.
Yeah.
But the sources we are diving into today, they completely shatter that firewall.
They really do. And to understand how they do that, we kind of need to move past that traditional simplistic view of genetics.
Yeah, let's upgrade the standard analogy. I like to think of it this way. If your DNA genome, the actual sequence of 3 billion letters, the A's, C's, T's, and G's, is a computer's physical hardware, the epigenome is the operating system.
I love that analogy. Your DNA is the hard drive. It's the silicon, the physical circuits.
Right, but the epigenome is the software that tells that physical hardware how to actually function. It dictates which applications, in this case, which genes to execute, which ones to compress, and which ones to completely quarantine.
And the key thing here is that the operating system is incredibly dynamic. I mean, every single cell in your body contains the exact same hardware, the exact same genetic code.
Which is wild when you think about it.
It is. The only reason a neuron looks and acts completely differently from, say, a liver cell or a white blood cell is entirely due to that epigenetic software.
So in the neuron, the software is basically opening the files for synaptic transmission and permanently locking the files that are meant for filtering blood toxins.
Exactly. And what the scientific community is now realizing is that this software is not just some static set of instructions that gets written once during embryonic development and then never changes.
Right. It's exquisitely sensitive to the environment.
It acts as a real-time data recorder. It's constantly patching and updating itself based on the nutritional, chemical, and psychological inputs that you experience throughout your entire life.
And the paradigm-shifting realization, the phenomenon that actually explains our whole cherry blossom thought experiment from the beginning, is that under very specific conditions, these software updates can be exported.
They can bypass that Weismann barrier entirely.
They get uploaded into the germline and then downloaded into the next generation, which brings us to this absolutely landmark 2014 study from researchers Brian Dias and Dias Ressler at Emory University.
This experiment is so rigorous and, frankly, so unsettling that it fundamentally altered the entire trajectory of epigenetic research.
Let's get into the setup.
Okay.
Because Dias and Ressler, they wanted to test if a highly specific learned fear could literally be inherited.
Right. And to do this, they used a system that is incredibly well mapped in mammals, which is the olfactory system. Smell, exactly. They took male mice and introduced them to a chamber. And into this chamber, they pumped a very specific odorant called acetophenone. And that has a distinct sweet smell, right?
Yes, it smells very similar to cherry blossoms.
But this wasn't some pleasant aromatherapy session for these mice.
Definitely not. Right as the acetophenone saturated the air in the chamber, the researchers administered a mild electrical foot shock to the mice.
Oh, wow. Okay, so this is classic Pavlovian fear conditioning.
Exactly. You pair a neutral stimulus, which is the scent of the cherry blossoms, with an aversive stimulus, which is the electrical shock.
And within just a few days, the mice completely learn the association.
Oh, yeah. After that, the researchers could just pump in the acetophenone without any electrical shock at all, and the mice would immediately exhibit a freezing behavior.
Their sympathetic nervous systems would just fully activate.
They were absolutely terrified of the smell alone. Now, because the olfactory system is so well mapped, we know exactly what is happening in the mouse's brain during this process. Right, because acetophenone binds specifically to an odorant receptor in the nose, and that receptor is coded by a specific gene called ULF151.
Right. And when the mouse learns to fear the smell, the brain actually utilizes neuroplasticity to adapt to the threat.
Dias and Ressler actually looked at the olfactory bulbs, like the physical scent processing centers in the brains of these traumatized mice, and they found a massive physical alteration.
It's incredible. The specific neural pathways, the glomeruli that are dedicated to processing that exact ULF151 receptor, had physically expanded.
The brain had literally built more neural real estate, more dedicated hardware, just to detect that exact cherry blossom threat.
The mouse is now hypervigilant.
Okay, but if the story ended right there, we would just have a really fascinating example of adult neuroplasticity, right? Like the brain change based on experience.
Right, which we already knew was possible. But Dias and Ressler took those traumatized male mice and they mated them with completely naive female mice.
Naive meaning females who had never been shocked and had never even smelled acetophenone.
Exactly. They produced a litter of pups, which we call the F1 generation.
And these pups, they grow up in a perfectly normal, totally stress-free environment.
But when the researchers exposed this F1 generation to the scent of cherry blossoms for the very first time,
the mice exhibited a heightened behavioral sensitivity. They startled way more easily. They showed fear.
They were terrified of a scent they had never encountered in their lives.
And they actually had more neurons dedicated to detecting it, just like their traumatized fathers did.
Now, this is exactly where skepticism naturally and rightfully arises in the scientific community.
Because the immediate assumption is, oh, we're just observing behavioral transmission, not biological inheritance.
Right, social cues.
Exactly.
Yeah.
I mean, if I'm terrified of spiders and a spider crawls across the floor, I might gasp, my body tenses up, or I grab my kid to pull them away.
Your child doesn't need to inherit a literal spider fear gene to learn that spiders are dangerous.
No, they just watch my panicked reaction and they internalize the threat. So the very obvious question with this study is, how do we know the dad mouse didn't just smell the acetophenone in the air, completely freak out, and then the babies observed him and learned to be scared?
Dias and Ressler anticipated exactly that critique, which is why the controls in this study are just so brilliant. First off, the traumatized fathers were removed immediately after mating.
So they never even met their offspring.
Never. There was zero opportunity for the fathers to model that fearful behavior.
Okay, but the mother is still there. Could the father's massive stress have somehow altered the mother's physiology during the mating process?
And like maybe his stress hormones triggered a subtle change in her behavior.
Yeah, making her a more anxious mother overall, which in turn just made the pups hyper-reactive to everything in their environment.
To eliminate maternal behavioral influence, the researchers utilized cross-fostering.
Oh, that's clever.
It is. They took the F1 pups born to these mothers and immediately transferred them to surrogate mothers.
And the surrogates were entirely naive, very relaxed females who had never interacted with the traumatized fathers at all.
Exactly. So the pups were raised by calm mothers in a completely calm environment. Yet, when exposed to acetophenone,
They still froze.
they still froze.
Okay, so that totally eliminates parental rearing and social cues. But to truly prove that this is a biological software transfer, you kind of have to eliminate the physical act of mating altogether, right?
Yeah.
You have to isolate the actual germ cell.
Which is exactly what they did next. They moved to in vitro fertilization.
IVF for mice.
IVF for mice. They extracted sperm from the traumatized male mice. They used that sperm to fertilize eggs in a petri dish and then implanted those embryos into surrogate mothers.
So in this scenario, there is absolutely zero behavioral or physical contact between the traumatized male and the mother.
And zero contact between the father and the offspring.
The only possible bridge between the father's trauma and the offspring's existence was a single microscopic cell, the sperm.
And the IVF offspring still showed the exact same heightened behavioral sensitivity to cherry blossoms.
And the exact same enlarged anatomical structures in their olfactory bulbs.
It's staggering. They even bred the F1 generation to create an F2 generation, so the grandchildren, and the effect persisted.
The physical brain structure and the specific fear response were inherited across multiple generations.
This really forces us down to the molecular level because if the genetic code, the DNA sequence itself, hasn't mutated.
Right, because we know a few days of getting shocked on the foot doesn't rewrite the actual 3 billion-letter genetic sequence of the ULFR151 gene.
So how on earth is the sperm carrying this information?
To find out, the researchers used this technique called bisulfite sequencing on the sperm of the traumatized fathers. Let's break down how this works because honestly, it's crucial for understanding this entire field of epigenetics.
Bisulfite sequencing basically allows scientists to physically read the epigenetic software.
So when you treat DNA with a chemical called sodium bisulfite, any standard untagged cytosine, which is one of the 4 basic letters of DNA, gets converted into a completely different chemical called uracil.
But if that cytosine has an epigenetic tag on it, specifically what we call a methyl group, the bisulfite can't touch it.
The tag essentially protects it from the chemical conversion.
Exactly. So by sequencing the DNA before and after this chemical treatment, researchers can essentially see a barcode. They can see exactly which genes are methylated and which ones are not.
Now, what actually is a methyl group? This sounds complicated, but it's incredibly simple. It's just one carbon atom bonded to 3 hydrogen atoms.
But when this tiny little molecule attaches to the DNA strand, usually at a region where a cytosine sits right next to a guanine, which we call a CPG site, it acts like a physical roadblock.
It's molecular steric hindrance. The transcription machinery, like the cellular proteins that actually read a gene and turn it into action, they physically cannot bind to the DNA because that little methyl group is literally in the way.
It's like putting a thick piece of duct tape right over a specific sentence in an instruction manual. The gene is essentially silenced.
But conversely, when those methyl tags are removed, which is a process called hypomethylation, the tape is ripped off.
The gene is exposed, it becomes highly accessible, and essentially highlighted for maximum expression.
So when Dias and Ressler looked at the bisulfite sequencing of the sperm from those traumatized fathers, they found severe CPG hypomethylation specifically localized right at the ULA151 gene.
The silencing tags had been completely stripped away. The gene responsible for building the cherry blossom receptors was highlighted.
The environment had physically altered the epigenetic markings inside the germline. And remarkably, when they sequenced the sperm of the F1 sons, they found the exact same pattern of highlighting.
So the father's biology essentially assessed the environment, determined that this specific floral scent was a matter of life and death, and sent a highly targeted software patch down to the reproductive system.
It programmed the offspring to be born with their internal alarm systems already calibrated to this specific threat. I mean, evolution didn't wait thousands of years for some random mutation to confer an advantage.
No, it adapted in real time.
But, mapping the olfactory pathways of a genetically identical, completely laboratory-controlled mouse is one thing.
The real, and frankly far more terrifying, question is whether this incredibly precise molecular transfer happens in the chaotic, uncontrolled, and messy environment of human history.
Exactly. We aren't dealing with sterile chambers and isolated scents. Human history is forged in war, displacement, systemic poverty, and famine.
The question is, do these massive societal traumas leave the exact same kind of highly specific epigenetic highlighting in our own human germlines?
And to answer that, we have to look at epidemiological data. And tragically, the 20th century provided several massive inadvertent cohort studies that actually allow us to trace these molecular scars.
The most heavily researched of these is the Dutch hunger winter.
The historical context of the Dutch hunger winter is so essential for understanding the biology here. So, in the winter of 1944 to 1945, the western part of the Netherlands was still occupied by German forces.
Right. And in an attempt to aid the advancing Allied troops, the exiled Dutch government called for a national railway strike. The goal was to cripple German logistics.
But in retaliation for that strike, the occupying administration placed a strict embargo on all food transport into the western provinces.
What followed was just a catastrophic, completely engineered famine.
A modern, developed, industrialized nation was suddenly plunged into medieval-level starvation.
The official daily rations dropped rapidly, eventually plummeting to below 500 calories a day.
The population resorted to eating grass, sugar beets, and famously tulip bulbs, literally just to survive the freezing winter. Over 20,000 people died of starvation before the country was finally liberated in May 1945.
It is a profoundly dark chapter of history. But epidemiologically speaking, it presented a deeply unusual scenario for researchers.
Right, because famines are usually protracted, they're geographically messy, and they're very poorly documented.
But the Dutch hunger winter had a distinct, sharp beginning and a very distinct end.
And furthermore, it occurred in a country with a highly sophisticated healthcare infrastructure and meticulous centralized birth registries.
This meant that decades later, researchers had this perfectly defined cohort. They knew exactly who was in the womb and at what exact stage of gestation during the 5 months of severe starvation.
So researchers like L.H. Loomy at Columbia University and Bastian Heymans at Leiden University realized they could track down these individuals who are now in their 50s and 60s to study the long-term biological legacy of that prenatal famine.
And the methodology here was incredibly robust. They didn't just compare the famine-exposed cohort to the general population.
Which would introduce countless confounding variables, right?
Exactly. Instead, they compared them to their own unexposed same-sex siblings who were conceived either before the famine began or after the food supply was completely restored.
That is brilliant. You are looking at individuals with the same parents, the same genetics, raised in the same households, the only difference being the nutritional environment they experienced during very specific trimesters of fetal development.
And when they drew blood from these individuals 6 decades later and analyzed their epigenomes, what did they find?
Well, they found profound, persistent epigenetic differences.
The individuals who were exposed to the famine during early gestation, so the first trimester, had significantly altered methylation patterns compared to their own unexposed siblings.
And the researchers zeroed in on a specific gene called IGF-2, which stands for insulin-like growth factor 2.
Let's explain IGF-2 for a second. In a developing fetus, IGF-2 is a critical driver of cellular growth and division.
It is heavily involved in how the body partitions nutrients and builds internal organs.
In the famine cohort, the IGF-2 gene was hypomethylated.
Meaning the silencing tags were reduced, the gene was more active?
Now think about the mechanics of pregnancy here. The placenta is not just a passive tube providing oxygen, it is an active sensing organ.
Right. It is constantly detecting the amino acid and glucose levels in the mother's blood.
So the intrauterine environment is sending a very loud biochemical signal directly to the developing fetus. The outside world is completely devoid of calories. You're going to be born into a harsh, starving environment.
So the fetus's epigenome reacts. It engages a survival strategy. Biologists actually call this the thrifty phenotype.
The software updates to fundamentally alter the metabolism.
It programs the body to hoard every single calorie it ever encounters, to prioritize fat storage over lean muscle mass, and to alter insulin sensitivity just to survive a resource-poor world.
The fetus is literally building a body designed for a famine.
But here's where the absolute tragedy of the mismatch theory occurs. Because the famine ended.
The Allies liberated the Netherlands, massive food shipments poured in, and the dietary landscape eventually returned to a modern, caloric abundance.
These babies, whose biology was meticulously, painstakingly optimized for starvation, were born into a world of plenty.
Their operating system was programmed for a reality that simply no longer existed.
And the consequences of that were devastating. By tracking this cohort over decades, researchers found that the individuals exposed to early gestation famine experienced significantly higher rates of obesity,
Type 2 diabetes.
elevated LDL cholesterol, and cardiovascular disease compared to their siblings. They even had a higher overall mortality rate.
The transient, temporary trauma of a 6-month starvation left a permanent molecular scar that relentlessly dictated their metabolic health 60 years later.
And as we dig through the rest of the sources, we see this isn't just an isolated, localized anomaly.
No, if we scale up to look at the Great Chinese Famine, which took place from 1958 to 1961, we see the exact same biological mechanisms playing out across millions of people.
This famine, which was driven by the systemic agricultural policies of the Great Leap Forward, resulted in tens of millions of deaths.
And the epidemiological studies from the Chinese cohort closely mirror the Dutch findings, but they actually expand the scope of the genetic targets.
Researchers found that early life exposure to the Chinese famine was associated with hypermethylation in a different critical metabolic gene, the INSR gene.
Which codes for the insulin receptor?
The insulin receptor is the literal lock on the outside of your cells that insulin must open to allow glucose inside for energy.
So if that gene is heavily methylated, meaning it's silenced, your body cannot build enough locks.
You develop severe insulin resistance. Your blood sugar stays toxically high. So again, a massive increase in metabolic syndrome directly tied to an epigenetic response to historical trauma.
But the research into the Great Chinese Famine also pushes the boundary into a much more complex system than just metabolism. It looks at neurodevelopment.
The sources explicitly note that prenatal exposure to this severe malnutrition is strongly associated with a multi-generational increase in cognitive impairments.
And a significantly higher risk of developing schizophrenia later in life.
This connects deeply to how a developing brain actually wires itself. I mean, building a human brain requires massive amounts of energy and highly specific micronutrients.
If the placenta is signaling profound metabolic stress, it forces the body to make trade-offs.
The fetal brain development is subtly but permanently altered. The neural pruning processes, the formation of synapses, the structural integrity of the prefrontal cortex, all of it is compromised.
Setting the stage for severe psychiatric vulnerabilities decades down the line.
Which brings us to perhaps the most sensitive and hotly debated frontier of transgenerational epigenetics.
Right. Because the Dutch and Chinese famines brilliantly illustrate the epigenetic transmission of nutritional stress.
The biochemical pathway from a starving mother to a fetus makes intuitive mechanical sense.
But what about pure, sheer psychological terror? Does the experience of extreme emotional trauma, violence, and fear leave a molecular legacy?
To explore this, we turn to the groundbreaking work of Dr. Rachel Yehuda at the Icahn School of Medicine at Mount Sinai.
Dr. Yehuda has spent decades studying the neurobiology of trauma, focusing intensely on Holocaust survivors and their children.
And I want to take a moment here to be very clear with you, the listener, because when we discuss events like the starvation of the Dutch, or the policies that triggered the Chinese famine, or the unimaginable systematic atrocities of the Holocaust, we are navigating deeply painful historical and political realities.
Right.
Our role here on the deep dive is to maintain a rigorous, impartial scientific lens. We are exploring the biological data found in these peer-reviewed sources. The historical analysis and the ideological debates surrounding these tragedies are vital, but our focus today is purely on the molecular reality of what this level of trauma physically does to the human organism.
And the biological reality that Dr. Yehuda uncovered is profound. She established a study involving cohorts of Holocaust survivors.
These were individuals who had been interned in concentration camps, who had witnessed systematic murder, endured torture, or survived by hiding in constant terror for years.
She evaluated their psychological health, their stress hormone profiles, and their epigenomes. And then, crucially, she did the exact same evaluations on their adult children.
These are children who were born after 1945. They did not experience the Holocaust firsthand.
They did not suffer the malnutrition of the camps or the acute threat of violence.
Yet the data showed an unmistakable pattern.
Yehuda found that the adult children of Holocaust survivors had a statistically higher likelihood of developing stress-related psychiatric disorders, specifically PTSD and depression, compared to demographic control groups whose parents lived outside of Europe during the war.
They exhibited an altered baseline of stress reactivity. Their internal alarm systems were literally calibrated differently.
And to understand why, Yehuda looked at their epigenomes, specifically targeting a gene known as FKBP5.
This is where the biology gets incredibly fascinating. What exactly is the role of the FKBP5 gene?
FKBP5 is a crucial regulator within the hypothalamic pituitary adrenal axis, the HPA axis.
Which is the body's central stress response system.
Exactly. When you encounter a threat, your brain signals your adrenal glands to release a massive flood of cortisol.
Cortisol accelerates your heart rate, floods your muscles with glucose, and physically prepares you to fight or flee.
But equally important to turning the stress response on is the ability to turn it off.
Right, because if you stay flooded with cortisol forever, your immune system crashes, your memory degrades, and you suffer massive systemic damage. You desperately need a negative feedback loop.
Exactly. Cortisol eventually makes its way back to the brain, where it binds to glucocorticoid receptors. When enough of those receptors are activated, the brain says, okay, the threat is handled, and shuts down the cortisol production.
So where does FKBP5 come in?
The FKBP5 protein essentially acts as a bouncer at the club for these receptors. It binds to the glucocorticoid receptor and decreases its sensitivity to cortisol.
It actively impedes the feedback loop, keeping the stress response going longer.
So Yehuda found that Holocaust survivors had altered methylation patterns on the FKBP5 gene. Given the unrelenting, prolonged trauma of the camps, their biology adapted by fundamentally altering how they process stress hormones.
But the real breakthrough was looking at the children. Yehuda found epigenetic changes in the exact same specific intron region of the FKBP5 gene in the adult children of the survivors.
The children were born with a pre-adjusted stress response system.
Their biological capacity to regulate cortisol had been fundamentally shifted by an event that occurred before they were even conceived.
Just like the mice and the cherry blossoms, the extreme environmental pressure forced an adaptation. The body attempted to prepare the next generation for the horrific world it believed they were going to inhabit.
This brings us to the most vital mechanical question of this entire deep dive. It's the hurdle that made scientists reject transgenerational epigenetics for so long.
We have established that the Weismann barrier is broken. We see that mice inherit fear pathways and humans inherit metabolic and stress responses.
But mechanistically, how is this actually possible?
That is the million-dollar question.
If the mouse's brain smells the cherry blossom, or the human mind experiences the terror of a war zone, how does that localized neural experience travel through the body, bypass the immune system, enter the reproductive organs, and physically rewrite the epigenetic software of a sperm or an egg?
For decades, this was the missing link. Without a known biological transport mechanism, epigenetic inheritance just sounded like magic. Or pseudoscience.
But the modern sources we are unpacking today have finally illuminated the map. The secret lies in a microscopic communication network that we entirely ignored for years.
Circulating extracellular vesicles.
Extracellular vesicles, or EVs. Let's really visualize what these are, because for the longest time, when cell biologists looked through electron microscopes, they saw cells constantly budding off these tiny nano-sized bubbles bounded by a lipid membrane.
And the assumption was that this was just cellular waste disposal. The cell was basically taking out the trash.
It was a massive underestimation. We now know that extracellular vesicles are not garbage bags. They are an extraordinarily sophisticated, highly targeted internal postal service.
Almost every cell in your body secretes EVs. They are essentially biological USB thumb drives.
A cell will meticulously package specific molecules, proteins, lipids, and highly specialized genetic material into a little lipid bilayer envelope.
And because they have this lipid membrane, they are completely protected from the enzymes in the blood that would normally chew up loose genetic material.
The cell ejects this secure data packet into the bloodstream, where it can travel vast distances, navigate to a completely different organ, dock with a specific target cell, fuse its membrane, and upload its payload.
It is a body-wide, real-time wireless network.
And the sources reveal that this communication network plays a shockingly vital role in reproductive biology, specifically during a process called epididymal transit.
Let's explore the anatomy of epididymal transit. When sperm are produced in the testicles, they're actually entirely immature.
They can't swim, and they don't have the biochemical tools to fertilize an egg,
To gain those abilities, they have to leave the testicle and travel through the epididymis.
which is a tightly coiled, incredibly long tube resting at the back of each testicle. Think of it as a specialized boot camp for sperm.
It takes roughly 2 weeks for a sperm cell to travel the full length of the epididymis. And during this journey, they're subjected to a massive influx of extracellular vesicles.
The cells lining the epididymis are constantly secreting highly specialized EVs called epididymisomes.
These epididymisomes physically fuse with the passing sperm, integrating their contents into the sperm cell.
And this right here is the breach in the Weismann barrier. This is exactly how the brain talks to the germline.
If the organism is experiencing massive systemic stress, whether it's the sheer terror of a predator, the metabolic panic of starvation, or extreme psychological trauma, the brain and the endocrine system secrete massive amounts of stress signals.
These signals alter the composition of the extracellular vesicles circulating in the blood.
Those system-wide EVs reach the epididymis, alter the local epididymisomes, and ultimately deliver the stress update directly into the maturing sperm.
The physical fusion of the lipid membranes is what allows the payload to enter. The sperm is literally updated while in transit.
But what exactly is the payload? What is the actual physical data inside these USB drives?
It isn't just a text file that says, "Fear cherry blossoms" or "Store more fat."
No, the primary active ingredients in this payload are non-coding RNAs.
We mentioned earlier that attaching a methyl group to DNA is one way to silence a gene. Non-coding RNAs are a second, incredibly powerful method of epigenetic regulation.
To appreciate this, we need to contrast it with something we are all much more familiar with now, messenger RNA, or mRNA.
Right. Thanks to the development of mRNA vaccines, we know that mRNA acts as the blueprint. It reads the DNA code in the nucleus, travels out to the cellular factory, and tells the ribosomes how to build a specific protein.
mRNA codes for something physical, but non-coding RNA, by definition, does not build proteins. So what does it do?
Non-coding RNAs are the master regulators of the factory floor. They control post-transcriptional expression.
Some of them act as interceptors. For example, microRNAs can physically bind to a strand of messenger RNA that is on its way to build a protein.
By binding to it, the microRNA either flags it for destruction by cellular enzymes or physically blocks the ribosome from reading it.
The mRNA is silenced before it can ever build the protein. Other non-coding RNAs can migrate back into the nucleus and recruit the actual enzymes that add or remove the methyl tags on the DNA itself.
So they are essentially the epigenetic management team. And the research highlights very specific types of these molecules inside the epididymisomes, particularly microRNAs and what are called tsRNAs, or tRNA-derived small RNAs.
Yes. The studies demonstrate that when an animal is subjected to severe stress, the specific profile and ratio of these microRNAs and tsRNAs inside the sperm changes dramatically.
When that sperm eventually fertilizes an egg, it is not just delivering half of the father's static DNA hardware.
It is dumping a massive payload of these altered, environmentally calibrated non-coding RNAs directly into the newly formed zygote.
Once inside the single-celled embryo, these non-coding RNAs immediately go to work. As the embryo begins to divide and differentiate into a fetus, these non-coding RNAs alter the developmental trajectory.
They act as an epigenetic memory, silencing certain growth pathways and highlighting stress pathways.
Guiding the structural development of the fetal brain, the HPA axis, and the metabolic system to match the precise environment the father just experienced.
The hardware remains untouched, but the software is profoundly rewritten from day one.
It is a breathtakingly elegant mechanism, and conceptually, it completely upends the traditional narrative of evolution.
It really forces us to reconsider a historical figure who has been used as a cautionary tale in biology classrooms for a century.
Jean-Baptiste Lamarck.
The Lamarckian redemption arc.
For those who might not remember their early biology classes, Jean-Baptiste Lamarck was an evolutionary theorist who predated Charles Darwin.
Lamarck's grand theory was the inheritance of acquired characteristics.
The classic textbook example used to mock him is the giraffe. Lamarck argued that a giraffe stretches its neck over its lifetime to reach higher leaves. Its neck physically lengthens a tiny bit, and it then passes that physically stretched neck down to its offspring.
Then Darwin arrived with the theory of natural selection. Darwin posited that evolution is driven by random, undirected genetic mutations.
A population of giraffes has varying neck lengths due to random genetics. The ones that happen to be born with slightly longer necks can reach more food, survive longer, and reproduce more, passing on those specific long neck genes.
Darwinism, eventually merged with Mendelian genetics, became the absolute bedrock of modern biology. Lamarck was completely relegated to the dustbin of bad scientific ideas.
Because logically, you can't pass down an acquired physical trait. If I lose my arm in an accident, my child is still born with 2 arms. If I spend 10 years at the gym building massive biceps, my baby is not born with a 6-pack. Acquired traits end with the somatic cells.
Exactly. But what we are unpacking today with epigenetic inheritance is fundamentally a molecular form of Lamarckism.
It is the literal inheritance of acquired characteristics.
The father mouse acquired a learned fear of cherry blossoms through an environmental experience, and he passed that specific acquired adaptation to his offspring.
The environment directly instructed the phenotype of the next generation.
So does this mean Lamarck was right all along? Are we discarding Darwinian natural selection?
Not at all. The modern scientific consensus is not replacing Darwin with Lamarck. It is synthesizing them.
The sources we are studying describe a deeply complex evolutionary framework on epigenetically facilitated mutational assimilation.
Which is a cornerstone of the inclusive evolutionary synthesis. And honestly, the best way to visualize this is as a relay race.
Let's break down the relay race model. Why does evolution even need a relay race?
Evolution has a massive timing problem. Environments can change incredibly fast.
A sudden catastrophic drought, the introduction of a novel predator, a rapid shift in temperature, or a global viral outbreak.
Darwinian evolution, relying entirely on random genetic mutations, is excruciatingly slow. It can take hundreds, sometimes thousands of generations for a beneficial random mutation to randomly appear, confer a survival advantage, and propagate throughout an entire population.
Right. If a new predator moves into your forest tomorrow, you cannot wait 3,000 years for a random mutation to give you better camouflage.
Your entire species will be eaten this week. You need an immediate solution.
Which is where the relay race begins. The first runner holding the baton is behavioral adaptation.
The organism changes its behavior. It hides during the day, it alters its diet, or, like our mouse, it quickly learns to fear the smell that precedes a shock.
But behavioral adaptation is fragile. It has to be learned from scratch by every single individual.
So the first runner passes the baton to the second runner, epigenetics.
Yes. The intense behavioral and environmental stress triggers an epigenetic change. The organism updates its software.
Through the mechanisms of extracellular vesicles and non-coding RNAs, it passes this updated software to its offspring.
The next generation is born pre-adapted. They don't have to learn the fear. The hardware is already highlighted for it.
This epigenetic mark stabilizes the new adaptation across a few generations. It acts as a biological stopgap. It buys the population time to survive the immediate crisis.
But we also know that epigenetic marks are relatively transient. They are written in pencil, not ink. They can fade over several generations if the environmental pressure disappears.
So they can't be the final solution. Who is the anchor leg of the relay race?
The anchor leg is pure genetics. This concept is often referred to as the Baldwin effect.
While the population is managing to survive the new predator or the famine, thanks to their temporary epigenetic software patch, random genetic mutations are still occurring in the background.
Eventually, purely by chance, a random genetic mutation arises that permanently hardwires that exact trait into the DNA sequence itself.
And because the entire population's survival is currently dependent on that specific trait via the epigenetic patch, natural selection strongly and immediately favors the new genetic mutation. It is rapidly assimilated into the genome.
Precisely. The epigenetic software patch keeps the computer running, keeping the species alive, just long enough for evolution to install a permanent hardware upgrade.
It bridges the critical gap between fast environmental shifts and slow Darwinian mutation.
This brings us out of the theoretical and into a very pressing, immediate reality.
We have talked about mice in cages. We have talked about historical famines from 80 years ago, but we are not just historical artifacts. We are living organisms.
And our current environment is actively programming the software of the next generation right now.
And the entire globe just experienced one of the most massive synchronized environmental shocks in modern history. the COVID-19 pandemic.
The pandemic provides a profound, real-time case study for everything we have discussed today.
Millions of people experienced a novel viral infection simultaneously. The immediate focus was rightly on acute survival, on lungs and immune responses.
But developmental biologists and epigeneticists immediately began asking a much longer-term question. What is the transgenerational legacy of this virus?
To explore this, we turn to a fascinating study conducted by researchers at the Florey Institute of Neuroscience and Mental Health, published in the journal Nature Communications.
They specifically investigated the impact of the SARS-CoV-2 virus on the male germline and the subsequent development of offspring.
The design of the Florey Institute study is critical because of what it chose to model. They did not model the severe, life-threatening cases of COVID-19 where patients are intubated in the ICU.
They modeled mild and even asymptomatic viral exposure in male mice.
This mimics the vastly more common human experience with the virus. They wanted to know if a mild bout of COVID-19, the kind you might just rest off at home, leaves an epigenetic footprint.
And what happens to the software after a mild infection?
They found that even a mild paternal infection significantly and durably altered the profile of small non-coding RNAs in the sperm.
The viral infection triggered an immune response, and that immune response cascaded down, altering the payload of the extracellular vesicles traveling to the epididymis. The sperm was repackaged.
The researchers then took these males, after they had fully cleared the virus and recovered, and mated them with naive females.
What was the outcome for the offspring who were conceived long after the virus was gone?
The offspring, who never encountered the virus themselves, exhibited significant behavioral changes.
Specifically, they demonstrated increased levels of anxiety and altered fear responses.
And interestingly, the researchers noted that this effect was sex-dependent. It manifested differently in male offspring compared to female offspring.
This sex-dimorphic response is actually a very common hallmark of epigenetic transmission, largely due to how the placenta interacts differently with male versus female fetuses.
Now, I have to pause and ask a challenging question here, because we need to differentiate between adaptation and damage.
Right.
When the mouse learned to fear the cherry blossom, passing that specific fear down made perfect evolutionary sense. It was a targeted warning system. Avoid this smell, it equals pain.
But a virus causing generalized anxiety in the next generation, is that an evolutionary warning?
Does being generally more anxious help a child survive a respiratory virus? Or is this just biological collateral damage? Is the virus just throwing a wrench into the machinery?
That is a brilliant distinction, and it lies at the very heart of the debate in transgenerational epigenetics.
Not all epigenetic changes are perfectly targeted adaptive warnings.
Some are indeed the result of severe systemic stress, simply destabilizing the delicate programming machinery.
In the case of the COVID-19 study, we are likely looking at a well-documented phenomenon known as maternal, or in this case, paternal immune activation, abbreviated as MIA or PIA.
Immune activation, meaning the epigenetic change in the sperm isn't caused by the physical spike protein of the virus itself, but rather by the father's own body reacting to it.
Exactly. When you are infected with a virus, your immune system launches a massive systemic counterattack.
Macrophages and T cells release a flood of inflammatory signaling molecules called cytokines. You've likely heard of the cytokine storm.
Molecules like interleukin-6 and tumor necrosis factor alpha flood the bloodstream.
These cytokines are absolutely essential for clearing the virus, but they are also highly toxic and incredibly disruptive to normal cellular function.
So this massive wave of systemic inflammation travels everywhere, crossing the blood-testis barrier, reaching the epididymis.
Yes. The sheer volume of inflammatory cytokines alters the environment where the sperm are maturing. The inflammation changes the epigenetic programming.
It's not a highly specific adaptive warning about a respiratory virus. It is a generalized, chaotic, inflammatory shock to the system.
And neurobiology tells us that generalized inflammation during germ cell maturation or fetal development profoundly alters how the fetal brain wires itself.
You can disrupt the migration of neurons and the formation of synapses, frequently resulting in a heightened baseline of anxiety, hyperreactivity, or cognitive deficits.
The implications of this are staggering. The pandemic isn't just a historical event that happened to us. It might be fundamentally shifting the baseline of human health for the next generation.
We might see an entire cohort of children born in the post-pandemic era who have a higher biological baseline for anxiety or neurodevelopmental challenges.
And it wouldn't be because of the psychological trauma of lockdowns or missing school. It would be due to the molecular footprint of their parents' immunological response to the virus itself.
That is exactly the sobering reality the researchers at the Florey Institute are pointing out.
If these findings hold true in human populations, and there is intense ongoing research to track this, it has massive implications for global public health and pediatric psychiatry.
It highlights that an infectious disease isn't just an acute crisis contained in a few weeks of illness. It has a long, silent tail that extends across generations.
I'm going to be completely honest, taking all of this in the deep historical trauma of the Holocaust, the lasting metabolic damage from the famines, the viral legacy of the pandemic, the fact that a simple shock can rewire your grandchildren's brains. It sounds incredibly bleak.
It paints a picture where you are just a walking repository of your ancestors' worst days, carrying their accumulated terror and viral baggage.
Are we just doomed? Is our software permanently corrupted by the tragedies of the past?
If the biological story ended there, it would be a story of pure determinism and a deeply pessimistic one. But it doesn't.
And this brings us to the most hopeful, empowering, and actively researched area of this entire field, the promise of plasticity.
The epigenetic erasure.
Exactly. This is the fundamental defining difference between a genetic mutation and an epigenetic mark. Reversibility.
A genetic mutation is written in ink. If the DNA sequence mutates from a C to a T, that change is permanent. You cannot think or exercise your way out of a genetic mutation.
But an epigenetic mark, the methyl group, the non-coding RNA profile, is written in pencil. It is highly plastic. The environment can write it, but a different environment can erase it.
To understand how powerful this erasure is, we have to look at one of the most foundational pieces of research in the field of behavioral epigenetics, the classic work of Dr. Michael Meaney at McGill University.
Meaney studied maternal care in rats, and his findings completely revolutionized how we view early childhood development.
Meaney observed that mother rats naturally display variations in how they care for their pups.
You have some mothers that are highly attentive. They spend a significant amount of time actively licking and grooming their offspring. We call this high LG, high licking and grooming.
This is high-quality, nurturing maternal care. And you have other mothers that are naturally more neglectful, offering very low levels of licking and grooming.
Meaney tracked the pups raised by both types of mothers. He found that the pups raised by the highly attentive, high-licking mothers grew up to be very calm, resilient, adult rats.
When introduced to a stressful environment, they explored confidently, their stress hormones spiked appropriately, and then quickly returned to baseline. They handled stress beautifully.
But the pups raised by the neglectful, low-licking mothers grew up to be highly anxious, easily startled, fearful rats with chronically elevated stress hormones.
Behavioral psychologists would just say, well, of course, good parenting creates well-adjusted offspring. But Meaney wanted to know the molecular mechanism.
He looked into the brains of these pups, specifically at the hippocampus, which plays a massive role in regulating the stress response.
He discovered that the attentive licking and grooming from the mother physically altered the pup's epigenome.
How does a mother's tongue alter DNA?
It's an incredible biochemical cascade. The physical, tactile stimulation of the mother's licking on the pup's skin sends neural signals to the pup's brain, triggering release of serotonin.
The serotonin activates a cascade of secondary messengers inside the neurons, which eventually turn on specific transcription factors.
These transcription factors physically recruit enzymes called demethylases to a very specific gene, the NR3C1 gene.
The NR3C1 gene codes for the glucocorticoid receptor. This is exactly the same receptor system involved in Rachel Yehuda's Holocaust trauma studies.
Yes, the enzymes recruited by the mother's licking actively strip the methyl tags off the NR3C1 gene. The gene becomes hypomethylated, completely highlighted.
Because it is highlighted, the pup's brain builds far more glucocorticoid receptors.
With an abundance of these receptors, the pup's brain is incredibly sensitive to cortisol. It detects the stress hormone immediately and efficiently shuts down the stress response.
The mother's physical care literally built a more resilient brain at the molecular level.
But Meaney had to prove that this resilience was actually caused by the environment, the licking, and not just inherited genetics.
It's entirely possible that calm mothers just have calm genes and they pass those genetic sequences to their pups.
To isolate the environment from the genetics, Meaney utilized cross-fostering once again.
He took pups born to the highly anxious, neglectful mothers, and on the day they were born, he immediately gave them to the calm, high-licking mothers to raise.
He also did the reverse, taking pups born to calm mothers and giving them to the neglectful mothers.
And what happened? Does genetics win or does nurture win?
The pup's destiny followed the mother who raised them. not the mother who birthed them.
The pups born with the genetics of the anxious mothers, but who received the high-quality tactile licking and grooming from the surrogate, grew up calm and resilient.
But here is the monumental finding. When Meaney looked at their brains, he found that the high-quality maternal care had physically erased the negative epigenetic programming they were born with.
Nurture physically edited the epigenome.
That is profound. The tactile sensation of feeling safe and cared for initiated a chemical cascade that literally removed the molecular scars of their lineage.
It proves that we are not locked in. The software can be patched, and the corrupted files can be deleted.
And this realization is currently reshaping the entire landscape of modern medicine.
If we understand that these epigenetic marks are driving aging, metabolic disease, and psychiatric vulnerabilities, and we know they are reversible, then we can actively target them. We're entering the era of precision epigenetic medicine.
This leads us to the concept of epigenetic clocks, which is popping up constantly in longevity research right now. Let's explain how these clocks work.
An epigenetic clock, the most famous being the Horvath clock developed by Steve Horvath, is a highly complex algorithm.
It looks at the methylation status of hundreds of specific CPG sites across your entire genome.
By analyzing this pattern, the clock can determine your biological age, which can be vastly different from your chronological age.
You might be exactly 40 years old chronologically, but if you have lived a life filled with chronic stress, poor nutrition, lack of sleep, smoking, or severe early life trauma, the epigenetic burden on your cells is massive.
Your software is degraded. The Horvath clock might look at your methylation patterns and determine that your cells are biologically functioning like a 50-year-old.
You are aging faster than the calendar.
But because these marks are written in pencil, because they are plastic, we can theoretically turn back the clock. We can erase the damage.
Precisely. The emerging field of geroscience is heavily focused on targeted interventions to reverse this epigenetic aging.
We now have robust data showing that environmental enrichment, rigorous cardiovascular exercise, dietary changes, and profound social support can literally alter your DNA methylation patterns. Cognitive behavioral therapy, by fundamentally changing how you process stress, can initiate the same biochemical cascades as the rat mother's grooming, altering the methylation of your stress receptors.
And it goes beyond lifestyle. Pharmaceutical companies are pouring billions into epigenetic drugs.
Yes. Researchers are developing targeted compounds like histone deacetylase inhibitors or HDAC inhibitors and DNA methyltransferase inhibitors.
These are drugs designed to enter the cell and actively strip away the aberrant epigenetic marks associated with historical trauma, accelerated aging, or even cancer.
We are moving from simply managing the symptoms of inherited trauma to actively rewriting the molecular code that drives it.
So, to bring it all together, we are not just helpless recipients of our ancestors' darkest moments. We are equipped with the biological mechanisms to edit the ledger.
Let's synthesize this incredible, sprawling journey we've been on today.
We started by looking at a mouse who learned to fear the smell of cherry blossoms, and we saw how that specific learned fear physically altered the neuroanatomy of its children and grandchildren, entirely transmitted through the epigenetic highlighting of a single gene within the sperm.
We then traced that exact mechanism out of the laboratory and into the messy, tragic reality of human history.
We looked at the agonizing legacy of the Dutch hunger winter and the Great Chinese Famine. We saw how severe starvation forced the fetal epigenome to engage a thrifty phenotype, adapting to a world devoid of calories, which resulted in a lifelong legacy of metabolic syndrome and cognitive vulnerabilities when those children were born into abundance.
We looked at the profound intergenerational scars of Holocaust survivors, seeing how sheer terror is written directly into the FKBP5 gene, fundamentally recalibrating the human stress response for the next generation.
We unlocked the mystery of how this transfer actually happens, discovering the microscopic postal service of extracellular vesicles, these biological USB drives, delivering stress-induced payloads of microRNAs directly to the maturing sperm during epididymal transit.
We saw how this intricate system rescued Jean-Baptiste Lamarck from the dustbin of history, serving as the vital first step in an evolutionary relay race that bridges the gap between fast environmental crises and slow Darwinian mutation.
We examined the very modern threat of the COVID-19 pandemic, looking at data from the Florey Institute showing how the massive immune activation and cytokine storm from a viral infection can alter the epigenetic payload of a father's sperm, potentially shifting the baseline of anxiety and neurodevelopment for an entire generation.
But finally, we found genuine hope in the profound plasticity of our biology.
We saw how a mother rat's attentive, tactile care can initiate a biochemical cascade that physically erases the epigenetic markers of neglect.
We saw how the modern fields of geroscience and epigenetic medicine are learning to read our biological clocks, utilizing everything from behavioral therapy to targeted inhibitors to wash away the molecular damage of the past.
The true takeaway, the deepest implication for you, the listener, is this. Your lifestyle, your environment, how you manage your stress, and the triumphs you achieve are not isolated events.
They are not contained just within your own physical body or your own timeline. You are engaged in a constant dynamic molecular conversation with descendants you will never meet. You are the current runner holding the baton in the evolutionary relay race.
And that leaves us with a final provocative thought to ponder. We've spent the last hour exploring how trauma, how the agony of famine, the terror of war, and the profound inflammation of a global virus can echo across generations, desperately trying to prepare our children for a harsh, unforgiving world. But the mechanism is entirely neutral. It records everything. So what biological legacy might we leave if we consciously choose to surround ourselves with joy? What specific epigenetic marks, what protective highlights do we write into our software when we immerse ourselves in intellectual enrichment, in art, in deep, loving social connection? What if the peace you actively make with your own trauma today and the deliberate happiness you cultivate tomorrow is the ultimate biological inheritance you leave behind? The next time you walk down the street and smell a cherry blossom, remember, you are not just experiencing a fleeting moment in time, you might just be writing the future. Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.